So for project two, I got a little bit behind, so we'll do it a little bit differently. Rather than having you put together a data set, I'm going to give you a data set to work with. You've already had some experience, at least a little bit, with downloading sequences from GenBank and how that's done in Mega. Just a bit of a reminder, there are many, many different programs. Some of them are software that you have to pay for. Others are freely available or are web-based. Mega, I use it because it's very flexible. It's not great at everything, but you can do a lot of things with it, and it's free. It works fairly well. And there are some downsides. That's kind of what comes with the territory when you're dealing with free software. But within those limitations, it does really a pretty good job. And so I'm going to start with this file. So I've got this large alignment. What I did was I chose one of the groups that we did for project one. These are dart frogs. You can look them up if you're interested. They're very cool group, fairly diverse. We found throughout Central America and South America, and an interesting group. And I've downloaded multiple species within different genera within the family Dendrobatidae, which is the poison dart frogs. And this is a 16S gene, so it's a ribosomal gene. It means it's non-protein coding. And ribosomal genes are interesting because they have these areas that are very, very conserved that makes alignment fairly simple for those regions. And then they have areas that are fairly diverse. And then they have these expansion regions which correspond to the loops in the three-dimensional shape of the ribosome. Overall, it makes them a pretty good gene to use because we have good alignment, but also diversity, and makes them a good gene to use for phylogenetics. Okay, so this is a fairly large data set. I put together 59 species here. And before we jump in and look at analyzing this data set, what I'd like to do is just talk a little bit about file format. You experienced a little of this, some of you did, on the first project because Mega saves it in their own file format. It's a .mas extension, and that can only be opened with a Mega program. It works well for that program, but there are other file formats that are more universally used that pretty much any program can open, and I'm going to look at two of them, and we'll just deal with the second one, the .fas, or what's called a FASTA or FASTA format. So the first format that I'm going to look at is called GenBank. And both GenBank, which is .gb, and FASTA.fas are simply text documents. But the GenBank format is a very data-rich format. There are multiple categories that can be used that can all be stored in this text format, and then different programs will be able to open it and utilize whatever data they need access to. So locus is a region. It gives information about the location on the chromosome, if there is, or the accession number. An accession number is a unique number that's assigned by NCBI when sequences are submitted to that database. And you've got lots of others or information here. The organism it comes from, its incomplete taxonomy, the title of the work or the paper under which that sequence was submitted, and then lots of other information. This is, again, the 16S ribosomal RNA. So you have all that information. So it's a really data-rich format and is really, really useful if you need to preserve lots of information about the sequence. GenBank is the way to go. Now, a simpler format is a FASTA format. And you can input some data into this, but you don't need to. So basically, FASTA is a bare bones, simple format. And the way it works is you have a first line that begins with this greater than symbol or caret symbol. After that, you can put whatever information you want in a long, long string of lines. So all I have is the genus and species here. But if I wanted to, I could include information about, and you can put spaces in or whatever, but 16S, and I could put the name, I could put lots of information here. And then this would all be uploaded as a name, but with that information embedded in the name. But for our purposes, we're simply going to put name. Maybe on this first one, I'll leave the gene also, just so we can do it. We can save it. So FASTA is a more universal program or universal format. Pretty much all programs can open FASTA format. Some people call it FASTA format. And it's just simply a text document. And this is the format which I'm going to give you this alignment in. So you're going to begin by opening this alignment in Mega. I've already got it open here, but let's close it and I'll show you how to do that. Okay. So reminder, you should be able to do the FASTA format because it is so universal. You can do it in just about any implementation of this software, this Mega software. So download the sequences. They're called 59 frog sequences.fas. So once you've done that, you open Mega, and this is the main Mega window. We're going to do, you can either do File, open a recently used file, or you can just do Align, open a saved alignment session. And then all you have to do is navigate to wherever you have it stored. So I've got it stored in this folder here under our projects. Project 2, here it is, 59 frog sequences. That's a .mas, but you can also, I think we might need to load it. So let me just show you how to load the .fas. So if we have a non-Mega format, then what we want to do is do edit build alignment and do retrieve sequences from a file. Okay, so we'll say okay, and then the same thing, we just navigate to where that file is. This is the easiest way for me to get there. Projects, project 2. And now we can see this .fas file. That's the standard one it uses. There are other formats. I don't know. They don't use GenBank here, but there are other formats also that are not really that critical for us. So here's our .fas format. We're going to open it up, and it brings it in this aligned format. Okay. Now, I want you to, once you've opened it up, kind of scan through it and just take a look at it. It's not a really long piece of the 16S. This gene is actually quite a bit longer than this, but we've got a chunk of it here. And again, you will notice areas that are very conserved, in fact, maybe completely conserved across this family. And then you've got areas where there's some variability, right? So we're looking in here, then this is really good data and information for relationships. And then we have areas where it's really maybe much more diverse, but you can notice that within certain species, there's genera, the Oophaga genus here, there's a lot of conservation. We can see they clearly stand out from the other ones. So notice again, even though you glance at it and say, oh, that looks very, very different, but when you look at them based on the genus they're in, we actually have a lot of conservation within those different genera. So this is really good, useful data and really shows a lot of shared characteristics between major groups. Okay, so we are going to show you how Mega is able to use some of these principles we've been talking about, how Mega helps us to select a model of evolution, and then we will do 3 very simple phylogenetic analyses and compare them. To complete this assignment, all you need to do is watch this video. You can pause it as you go, and I'd like you to follow along with the data set in Mega. So by this time, you should have opened this data set in Mega, and Mega will open 2 windows, right? So you've got your original main window here, and now we have an alignment window. And if we were to open, do a blast search or open another browser looking at the GenBank database, it would open a 3rd or 4th or 5th window. And that can be a little tricky for some users because most users are not used to software that opens multiple windows. So just be aware of that. It's just not a single window when you're using Mega. Okay, and we'll go back and forth, but I'll try to be clear about which window you're in so you don't get confused. So back to the alignment window here. Now what we would like to do is to be able to figure out what model of evolution is going to be best to do this, and then also the parameters. Okay. Now in Mega, the way that we do this is we need to activate this data set in the alignment. So to do that, we're going to go to Data, and then we're going to go to Phylogenetic Analysis. Now, if you've already loaded a window, it'll ask you, do you want to reload? But since we just barely opened this, it should be pretty straightforward. Now, the first question it says, is this protein coding nucleotide sequence? And for most data sets, your answer would be yes. But because we're dealing with ribosomal data, this is never translated into a protein. So we're going to say no, and that will limit the things that we can do. So we can't do things like look at translation into protein codes, because that's nonsensical for this data set. We aren't going to be doing analyses looking for strength of selection and other things that we'll talk about at later dates on amino acids, because there are no amino acids for this data set. It's just DNA that's transcribed into RNA. So we'll say no, and it will load it very quickly. And what you will notice if you look back at your main window now is we now have an extra little item here, which is our data set. And if I hit it, it's going to close it, and then we could open a new one. So we don't want to do that right now, but that means we have our data set looked at it. We can look at it in a different format, which is kind of a simplified alignment format. But again, that's not as important for us right now. So right now we have our alignment, and we've got our main window. Now, the first thing we want to do is look at a model of evolution. And so you've got this link in the main window called models. I'm going to click on that. And you have a couple of different options, some of which we'll talk about later. Many of these are a little bit more, and some of these are only for protein coding sequence. But what we want to do is find the best DNA or / protein model. And it will only let us do DNA models since we told it this was a non-protein coding data set. Okay, so we click on that. For this one, we can use our default. Now, the only changes you might want to make are if you think that these gaps are not meaningful. This is like, for instance, this is too divergent. You can actually get rid of gaps altogether. Or we can ignore the gaps and use the areas that have nucleotides listed in those gaps. But that can be problematic. So we're going to use all sites, including gaps. We're going to use the neighbor joining tree as the automatic beginning for this, and we're going to run an analysis. And this is a medium-sized data set. It doesn't take too long. And it's going to test all of these different models, and it does it with 8 different threads. So it basically tests them in different orders and compares them in a different order, because sometimes the order in which you compare them to one another can have an impact on the outcome. And then it gives you a readout. So let's just spend a minute looking at this readout. There's a lot of information here. But basically what it does is it lists from top to bottom the best model overall based on a couple of tests, a Bayesian information criterion and an Ikaki information criterion. Don't worry about those. We'll maybe talk about them very briefly later on. But all you need to know that these are statistical tests that try to find the best model of evolution. And the lowest score for these is the very best one overall that's decided by the software. And so notice that it has determined that our GTR, which is the most complex model, plus gamma, which is allowing rates of evolution to vary across different sites, plus a proportion of invariable sites, right? So GTR plus G plus I, that's been selected as our best model of evolution. So it's a complex model of evolution that's decided as best overall. And then it even gives you all of the values for the different parameters. So here's our proportion of invariable sites. Here's our gamma distribution, which allows for some amount of flexibility in the rate of evolution across sites. And it goes through all of these different sites. Now, you don't need to memorize these or write them down and to input them later, because it's a quick calculation for programs to do that. So most programs, including Mega, will calculate all of these for you as long as you just tell it, oh, I want to use a GTR model. Okay, so this is basically just to select our best model of evolution so we know what it is when we do the analyses later on. Okay, so I'm going to save that. We may come back to it, but all we need to do is remember we're using a GTR plus gamma rates, and then we need to make sure that invariable sites are used, which is done automatically in Mega. Okay, so we have done and selected our model of evolution, and the program has picked which one is best. Okay, so we'll just kind of put that to the side and keep that in mind. So the next thing that I would like to do is actually do a phylogenetic analysis of this data set. Okay, and again, there are different programs that can do this, and some are a little bit more powerful than others, but a little bit more or less user-friendly. Mega has its drawbacks, but it's fairly user-friendly, and the nice thing about it's very flexible. So we can do just about everything we want to do for this class with this one single program, which is why I've chosen it. Okay, so let's now do a phylogeny. We've already got our data set activated, so all we need to do is come to this option on the main window, again, not your alignment window, and select phylogeny. Okay, and we're going to do 3 quick analyses, and we'll save all of them and do a quick comparison at the end. The first one we're going to do is a very simple neighbor joining test. Okay, so neighbor joining, we'll leave everything at its default, and we'll just say OK. Now, neighbor joining analyses are very quick, and other analyses will be rather quick for this one too, because it's a again, medium-ish, but fairly small data set. And once you do analyses, it opens up this Tree Explorer window, it calls it. And there are different tools we can use here. I'm going to show you the different tools, and then we're going to do some side-by-side comparisons. Okay, so let's just scan over the results and see if anything jumps out at us. So the default for a neighbor joining tree is to show you a phylogram, which is where the branches estimate amounts of evolution. And if you go to the bottom, there'll even be a little bit of key about how many changes. We don't need to worry so much about that. But remember, these branch lengths are meaningful here. Okay, so let's scan down through it. It's a large-ish data set, so we'll look at it. But basically what I want to look at is for genera where we have multiple species represented, do we have a single monophyletic group represented in our analysis? So notice Anomaloglossus. And we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different species. 10, 11, 12, 13, 14 different species, 15. Wow, it's quite a bit. 15 different species of Anomaloglossus represented in this alignment. But notice they all are grouped together with a single common ancestor outside of all of the other species. So Anomaloglossus is a monophyletic group. And its sister taxus is this Allobates, the one that we put the gene reminder on. Now remember, this is an important, remember this name. This is an important one. We're going to come back to it. But let's look at other genera that have multiple representatives. Manofrini, and you can Google these if you want. They're pretty little frogs, but not important since we have the taxonomy here. Manofrini is, again, monophyletic with a single common ancestor. And notice that the common ancestor of Manofrini went through more evolution. This is a longer branch than the common ancestor of the Anomaloglossus. But still, that branch that separates the genus from the other ones is longer than these branches, most of the branches within the species. Okay, Ameriga, again, only 3 species represented, but a single monophyletic group. Colostethus, aren't these great names great? Colostethus, single monophyletic group. Now here's a little bit of an interesting one. The Silverstonia is not a monophyletic group. It has Dendrobates, which is monophyletic, but that is nested within the Silverstonia. Okay, so this is a little note we may want to make with a larger data set or with more representation to determine whether or not really this might tell us that Silverstonia is not a valid taxonomic group, that we need to include the Dendrobates as part of it. Again, usually with one single data set, we don't jump to conclusions, but it gives us a hint. Hyloxalis, monophyletic, but only 2 representatives. Phylobates, monophyletic. I only have a single representative of Menophrenia, so we can't determine whether it's monophyletic because we don't have multiple species. But all of the others, we're just going to go through here rather quickly. I think this is a mistype. Those should be the same genus, Excidobates with a C. Monophyletic, Dendrobates, again monophyletic. Delphobates, and then we have this larger genus represented, Oophaga, with some very short branches in between there. So our taxonomy here matches fairly well with our alignment based on our neighbor joining tree, which again is a fairly quick analysis. So I'm just going to move that to the side, and then let's do a parsimony analysis. Okay, so phylogeny, we still have our data set active here. And one of these is construct test maximum parsimony tree. Don't worry about test of phylogeny. We're going to use all sites again. And then these things down here are just how it searches. Some are a little bit more thorough than others, but we're fine with the defaults for the search strategy here. We're going to say okay. It's going to take a little bit more longer than the neighbor joining analysis, but not a whole lot. So let's make this one a little bit bigger, and then let's compare. Now notice the first thing that jumps out is that our Oophaga, which appeared down here at the bottom, are up here at the top. And this again is something that you need to remember. It doesn't matter where that branch occurs because we can shift things around. So this is a good chance actually to show you some of the tools and talk about how we view trees without changing relationships. And hopefully you're getting fairly comfortable with this. You remember some of this from our first unit. So we've got Anomaloglossus at the top of our phylogeny, and we've got Oophaga here. And if we find Anomaloglossus, it's going to be kind of nested down here in the middle. So if we want to compare these, it would be handy to kind of rearrange, switch without changing relationships, but just organize these in similar ways. And we can do that with tools here in Mega. So let's go off of, it doesn't really matter which one we go off of. Let's put anomaloglossus at the very top of our phylogeny if we can. Now, to do this, what we need to do is to be able to root the tree to our outgroup. Now, remember when I asked you to remember this species, Allobates? That is the closest relative to all of the rest of these, and we know that from previous analyses. We might need a selection of more distantly related. Maybe we could put a bullfrog and a toad and other things in there, but that work's already been done, so we know that this should be the root. And so one of the tools over here on your left-hand toolbar, we won't look at all of these, but we'll look at some, is this one here. And if you hover over it, it says root the tree on a selected branch. So I'm going to choose that tool, and then I'm going to come over here, and I'm going to pick this Allobates branch. And it will automatically reroot it so Allobates is at the root, and then everything else comes out. And let's do the same thing here. Let's find Allobates, pick our rooting tree, our rooting tool, I mean, and let's find Allobates here. I think it's going to be right there, and we're going to root it there. And so that right away will help a lot to organize these in a similar manner. So notice I've got Allobates, and then as we scan up, sometimes the size of these trees makes them a little trickier, but as we scan up, we've got the next group, which is Anomaloglossus genus, and then everything else comes out on the other branch from Anomaloglossus. So let's see if it's the same over here. And sure enough, here's Anomaloglossus, and then everything else is connected to the other branch. And we go up to the next one. We've got the Manofrini, all one group here. Manofrini, all one group, which then is connected to everything else. And I'm going through this rather quick, so if you're getting a little bit confused, don't worry. Now, notice we've got a little bit of, I guess it still is the same here. So our next group then, that is on this phylogeny, is a group that's composed of Epidobates, Silverstonii, Colosteus, and Amira, and the Hyloxus. Now, this is one where the parsimony disagrees a little bit with the neighbor joining. Notice that in this one, the Hyloxus, this one species, is more closely related to these other genera, and then this Awa species is more closely related to these. Whereas here, the Hyloaxus are in a single group. So that's a disagreement, a slight difference between the parsimony tree and the neighbor joining tree. Okay, so right away there's a little bit of a difference. Let's go up and look at this last major group. Philobates and Minobates. We've got Philobates and Minobates. And now we have, again, a little bit of a difference here because in this one, oops, sorry, I scanned a little bit. It's not a difference. So Exodobates here and Ninobates there, and then Rantiomaya. We'll look at one of these. There's some, these are pretty frogs, but we'll look at some just so you have an appreciation for them here at the end. Dendrobates is next, same. Adelphobates. Now notice something, and I didn't talk about this. The parsimony tree by default is not a phylogram. That although the branches, notice that they're all spaced in a nice even manner without differences in branch lengths like we see over here. And that's a very common way to present parsimony trees. The most common way. You can actually calculate branch lengths. It's a little bit of a lengthy process. We won't do it, but the Oophega here. So a few differences. And so if you were doing an analysis and writing this up, you might say, these trees are largely congruent. However, there are some disagreements with the placement of these species of Hyloxalis. It's hard to say, Hyloxalis. And parsimony has a paraphyletic, actually polyphyletic Hyloxalis because we have a branch over here and a branch over here we'd have to cut out to get them together. Whereas the neighbor joining tree puts them together. Okay, but other than that, it's largely congruent. So that would be a good talking point and something that maybe would need to investigate with a larger data set or further species representation. Okay, the last thing that we are going to do is our maximum likelihood. And what I'm actually going to do is let's close down the neighbor joining tree. We can just discard those. We don't need to save anything. And we'll leave the parsimony tree here, and let's look at our maximum likelihood tree. So to do that, we're going to come back here, and we are going to do a phylogeny. We're going to do a first one, construct or test a maximum likelihood tree. We're not going to do any testing right now. We'll talk about tests we can do at a later chapter. Okay. But remember that we want to pick the model of evolution. Now, depending on how you've used this program before, I think whatever one you used last comes up in this window. If it's not general time reversible, you need to use the drop-down menu and change it to general time reversible. And you'll notice some of these, there's some slight variations from the ones we learned, but that's the one we want. And then you need to also select, if you haven't, that it's gamma distributed rates, and it has invariant sites. And remember, we did both. Gamma distributed with invariant sites. So that's the option that we need to pick. Now, the number of discrete gamma categories is going to be 5. That just allows for the most flexibility across our range, our gamma distribution, which is the range of different rates that we can have at each site. Okay, we'll leave this at all sites. Tree inference, again, is how fast or how thorough it goes through searching for different trees. That's fine. We can leave it that way. Now, maximum likelihood test is a little longer than a parsimony, but I picked this data set so it's not prohibitive. You're not going to have to wait for a long, long time for this analysis to be completed. So let's do it. So again, it only took a few seconds for it to complete this one, maybe a third as long for the parsimony. And as we add more species, that jumps up very, very dramatically. So if I had like 75 species, it might take 20 minutes to do your maximum likelihood analysis. We're only dealing with 59. And if I had 100 species, it might take 4 or 5 hours, or we might have to start using cloud computing resources. Now, the default, again, is to show this as a phylogram. If yours isn't showing as a phylogram, this little option over here actually toggles back and, it's this one, toggles back and forth between a cladogram showing only relationships and a phylogram showing relationships and branch lengths. Now notice the branch lengths within Oophega are very, very short for most of the species, with this one being the most divergent. Okay, but the rest of them are fairly short, meaning there's not much difference between them. In fact, some of them are shown as polytomies. And notice, I think we had some other polytomies down here also. Yeah, so notice these 3 species, these 2 species, and then the Dendrobates, Adelphobates, Oophega group are all represented as a polytomy. 1, 2, 3 branches, all coming from that same ancestral branch or ancestral node. So there's not a lot of difference at some of these deeper branches, and the maximum likelihood couldn't quite distinguish between them. So let's do a comparison. And again, to do this, we want to root to the same one. So let's go find our root here. There it is, Allobates. I'm going to select the rooting tool and then select that branch. Sorry, rooting tool. This is just to select different items in the tree, and then once they're selected, you can implement the different tools there. So I did it kind of backwards there, but that's fine if you want to. You can select a branch and then root. So let's compare the parsimony tree to our maximum likelihood tree. We've got Anomalglossus, that large multiple represented, but monophyletic group, monophyletic group. It then is sister taxon to the rest of them. And then we have Manofrini. And even we could look at within the genera. I've kind of been skipping over within the genera, but notice within the genera, all the relationships are the same there for the Manofrini genus. We don't have time to look at every single one of these, but we'll do a couple here. Now, this one isn't interesting. When we used a model of evolution, we got a slightly different result here. And remember, even in our neighbor joining, I believe that these Silverstonia were paraphyletic. Okay, and also in the parsimony. But in the maximum likelihood, we have a single monophyletic Silverstonia. So in this case, the relationships within that genus are sensitive to different methods of phylogenetic inference. Epidobates looks good, looks the same. Cholestis, there we go, looks the same. The next one up is Amiriga. There we go. Now here we have another difference. Remember that we put this Hyloxalis in 2 different branches in the parsimony analysis, but here they're in a single group that is sister taxon to all the rest of these species up here. And then the rest of it, Phylobates, Miniobates looks the same. Exitobates, again, I misspelled one of the genera there, but those are the 2 in the same genus. Adenobates, Rantiomyia, Dendrobates, and notice the branch length here is very, very short. You can hardly even see it. So not a lot of evolution between the separation of those species, of those ancestors. Dendrobates, everything else looks pretty much identical. And I'm not looking real carefully at relationships within the genera, but we could do that. Let's just do it for this last one. So in this last one, we have Granulifera as the sister group to all the rest of the Oophaga. The next one here in Parsimony is Pumilio, but it's different down here. So there's a different set of relationships within this genus for Oophaga versus maximum likelihood. And so those are the things that you could begin to investigate. All right. So I've just shown you some of the tools that we can use. I'll show you a couple of others here that you might want to use. We're not going to get into this a whole lot. You're not going to be tested on this, but it might be handier just to think about and to illustrate some of the principles. So in addition to rooting, we can also swap selected subtrees. So I can pick this part, and when I do that, all it does is switch it around. So notice Pumilio's here at the bottom. And now Polymio, these have been moved around, but without changing relationships. All I'm doing is switching the branches. And I'm going to show you one last thing here before we go. Sorry, hover over that. We can flip this. Oh, that's the one that I wanted to show you. Flip the selected subtree. So Pumilio's at the bottom. Now it's at the top. But we're not changing relationships. We're just simply organizing it in a way that might be easier for you to read. You can also do a collapse. So if we wanted to talk about just the genera, what we could do is come to these common ancestors for each genus, and you have different ones. We could say, oh, this is my Oophaga group. We'll leave everything else on the default, but we could, well, let's go ahead and change the color. It's kind of handy to do. Okay, so we'll say okay. I didn't like changing the color. So anyway, that now represents the entire Oophaga group, and you could do that for each of your genera. Let's see if that was a general one. This is Dendrobates. Okay. Yeah, this is occasionally you get these little weird errors. Again, that this is not as stable, but so we may have to label these or write down what they are. But we remember, or we can label the different genera, and so maybe come up with a summary tree, and the width of these represents how many species are represented in each genus. Okay, there are other tools. If you want to play around with them, great. If not, don't worry. We're not going to go too much about these. Now, the last thing that I want to do is just to give you kind of an appreciation of this group that we're looking at. Let's take a look at some of them. So Oophaga literally means the egg eaters, and because the female, after she, and this is a fairly diverse group with lots of color variation, the female, after she lays an egg up in the like a small pool of water in a tree in the rainforest, she will then feed her babies by laying infertile eggs, and then the babies eat those eggs. So the mother actually takes care of her babies in that way. Dendrobates is another interesting group. Very colorful frogs. They do exude toxins. That's the name, the common name, poison dart frogs. Again, fairly diverse. The last one I'll just show you just so you get appreciation. Rantiomyia. I like this one. It kind of looks, to me, I always call it like the Spiderman Frog. Blue on the bottom and red on the top. Anyway, they're kind of fun frogs. They're rather small but very bright and colorful, and they do exude toxins. When they're kept in captivity, they usually don't have toxins. They have some interesting reproductive strategies. Here is a male Rantiomyia with a tadpole on its back. They carry them around and can move them from place to place if necessary. So an interesting group. And here are their genes. We're looking at their relationships. So kind of a fun thing to do. Now, to complete this assignment, to just ensure that you've actually done it, there will be a quick, we'll call it questionnaire, that you need to fill out. And it will go over some of the topics that we talked about. Fairly simple questions. If you've watched this video, they should be straightforward. For this project, all you need to do is go through this, complete the questionnaire, and then just submit that assignment.